Good morning. ISR is a spin-off company from Karolinska. We're a life science company in phase 2 clinical trials, and we are about to celebrate our third year being listed at First North Nasdaq. ISR is not focusing as many other drug companies, where they are focusing on the virus, the bacteria, or the tumor. ISR is focusing on the host. We are developing a set of drugs that will stimulate the immune system. The immune system will then target the virus, the bacteria, the cancer. Our first goal is to work with HIV. HIV, as you know, is a huge problem worldwide, where more than 37 million people are infected. 35 million people have died so far, and about 1 million people die every year. Although we have wonderful drugs that, if you take them properly, will give you a nice, high quality of life, you can never be cured. Our entry points is with an understanding from cats suffering from FIV, the cat equivalent. Colleagues of mine castrated cats on their street on an epidemic, and they survived. They cleared the virus. And years of research has demonstrated that it is a compound GnRH, gonadotropin releasing hormone, that is produced and causes effects that cures the cats. So when we stimulate the immune system with GnRH, T cells that carry the virus, this HIV virus, will be recognized, killed, and eliminated. It is like raising a plague flag. So an immune cell that is infected with the HIV virus, when stimulated with GnRH, it will start to upregulate flagpoles, class 1 molecules, carrying the HIV flag, which can then be recognized by killing cells and thus eliminate the disease. Carrying out a clinical trial in South Africa, we have demonstrated that using GnRH is safe, very well tolerated, and it diminishes the viral load of the HIV virus. Again, it stimulates T cells, and it stimulates specific T cells that recognize the HIV virus. What became very interesting is that after 4 weeks of treatment, we had a follow-up period for a month, and then the effect continued. So viral loads went down and specific T cells increased. That suggests that the method we are using causes a state of vaccination for the patients. And that is in sharp contrast to what you see when you stop ART medication. Within 10 to 12 days, the virus skyrockets again. So this leads us to another question. Can HIV be cured? Yes, there are 2 cases that have been described where you have cure. And that is because the virus has a certain entry point. It uses proteins on the surface of T cells. And 2 patients that needed a stem cell transplantation for other reasons, also having HIV, the stem cell match was chosen that one of these entry points was mutated, inhibiting the entry of the virus. So the virus could no longer enter into the cells. And by wiping out the immune system carrying the virus, they were cured. However, unfortunately, this is something that's going to be available for most patients. That's going to be a small, minute fraction. So what is the challenge with HIV? HIV is carried in our genome. So within our T helper cells in our immune system, we have a mole. The HIV virus inserts its DNA into our chromosomes. And that means that the disease is lifelong. This is something we are trying to address right now in a clinical trial here in Stockholm and in Gothenburg, ISR 03, where patients will be randomized either to be followed when they are on perfectly well-designed ART regimens and their virus loads cannot really be measured, or they will be treated with GnRH during 3 months and then a 3 months follow-up. The study is going on now where we are recruiting patients, and more than half the patients have already been evaluated. recruitment, as in many clinical trials, has been a little bit slower than anticipated, but one has to realize that many of the patients are completely well, and their desire to participate, coming to the doctor 8 times, getting injections just for a clinical trial, is less than anticipated. But we are getting there, and we are actually recruiting additional sites to get the study finalized. Another challenge with HIV is ART resistance. Like bacteria, the HIV virus can develop a resistance, so it no longer is responding to the treatment. WHO came out with an alarm report 2018, naming that 10% of the population infected have ART resistant. 1 in 10 being newly infected will actually be infected with a resistant strain. Women that start their first line of ART treatment are twice as likely to have a resistant strain. And what is even more concerning is that children born to women infected with HIV have a very high risk of getting a resistant strain. And that means that you have a very high risk of developing AIDS. It's forgotten almost by a generation that 20, 30 years ago, getting an HIV diagnosis was the equivalent of death. That was a death sentence. So we are now setting up a collaborative program with Berlin. So colleagues in Berlin will conduct a study on ART-resistant patients. A small study on 15 patients, where we during 6 months will treat them with GnRH and follow up how we can eliminate mutated virus and making the patients again susceptible to ART. To remind us about the size of the market, we can see that it's a huge market for HIV drugs, and it's predicted to, in the next 6 years to come, to almost double. And of course, here we believe that we can contribute with an additional set of an immunostimulatory drug that will help in the fight against HIV. The company has an additional line of work where we have our own compound, which has been a modulation of an existing antibacterial drug, azithromycin. By modulating azithromycin, we now have an immunostimulatory compound where all the antimicrobial activities has been eliminated. And this is something we developed for cancer and for hepatitis B. So in our daughter company, Immune System Regulation Oncology AB, it's a spin-out completely owned by the mother company, where immuno-oncology is the highlight. Here we are working together with checkpoint inhibitors to make a new combinatory drug for therapy of solid tumors. The strategy is to build to phase 1, phase 2 clinical trials and then join forces with big pharma in the field of CPI. Checkpoint inhibitors was awarded the Nobel Prize 2018. And that was because 10 to 20% of patients with solid tumors such as malignant melanoma, non-small lung cancer or urinary bladder cancer responded very well. However, 80 to 90% of the patients do not respond at all. And that is because tumor cells are smart. They need to avoid the immune system. And here is the point where we hope to add to the knowledge of 10 to 20% with a complementary effect, where ISR 50 is an immune stimulatory that would help and enhance the immune recognition of the T cells. Carrying out studies in mice in melanoma, which is a very, very difficult tumor form, we can demonstrate that we add to the checkpoint inhibition function. So another 10 to 20% of effect can be seen in mouse models. So for our ISR oncology program, we are now walking into the immuno-oncology field, another field where there is a tremendous increase to be expected the next coming years. The majority of the growth will be in the immuno-oncology field, where the majority is with immunocheckpoints and immune system modulators. and that's exactly the field where we are in. The ISR 50 molecule seems also to be very useful for hepatitis. Hepatitis B is a disease that is even larger than HIV. More than 300 million people are infected, and almost 1 million people dies every year. Next to smoking, HPV is the second most causative known agent to cause cancer, and this is liver cancer. So our ISR 50 has a unique position because we stimulate a particular cell, the Kupffer cell of the liver, which is the immunostimulator of T cells. And by providing ISR 50, we will stimulate the patient's own immune system to be able to recognize infected liver cells and eliminate the B cells. There's a huge unmet need. There are no drugs really available. There are some small signs that you can inhibit and postpone the growth, but there is no cure whatsoever. And we believe we have a position here where we can prevent the development of cancer and treat both acute and chronic hepatitis. Our strategy in ISR for HPV is to move our activities to China. We will focus on the biggest market in the world. In Asia, more than 60-70% of all cases exist. We know that this market will grow, and it's expected to grow with about 4.4% of annual growth rate to reach a market of about 1.5 billion US in the next years to come. Our business model is now to work with and discuss with companies for a license for the People's Republic of China market, or discuss direct investment in our daughter company, or probably and likely a combination of both. So ISR 50 is then our lead IND product, and that will be used in an injection format for oncology, and it will be used in a tablet format for HPV. We have now developed the first batch of our GMP, and the next one, the larger, a couple of kilograms, will be delivered in February. We have made a collaboration with InnoStar, which is a large, very, very active CRO company with lots of connections to the industry in China for the final end of the toxicology program, where we will be able to, by the end of the summer, have a finalized report that then will be used for an application for the Chinese FDA to conduct clinical trials. And we hope that they will be able to start in the beginning of next year in China. So with this short overview of ISR Holding AB, I've been telling you that we actually have 3 lines of investigations that are ongoing. HIV, where we have a phase 2A clinical trial that is presently on its end stage, where we hope we will have the results during the year. We will initiate a niche indication of art resistance starting during this year in Berlin. And we're developing our IND product that will be ready to be tested in first-in-man studies in HPV in China and in Europe, preferably here in Stockholm for oncology, where urinary bladder cancer will be our first indication. Late-stage urinary bladder cancer, where you have spread disease, where you have an expected life of about 6 additional months. So thank you very much, and I'll be happy to take questions.
Thank you very much, Ola. Interesting presentation. I'm actually going to turn to the audience first of all to see if there's anyone who has any questions here. And if you do, I have a microphone that you will have to use in order to have our online viewers also hear the question. If no one has any questions, I'll go ahead and ask a question then. I think that the status of the HIV study, the timeline there, what can you tell us about that? How you had some trouble with the patient recruitment earlier last year, and what's the status right now?
The challenge is whenever you're going to do a clinical trial that you have a very, very tight description in order to be included. So we have included more than half of the patients, that is approaching 30 patients. But we also have 19 failures. They were interested to participate, but they didn't fit the criteria. So they need to have been during treatment for more than 3 years. They can never have been low in CD4 cells and so forth. So that is a challenge for us to actually find. I mean, we are blessed in Sweden. There are so few patients that have HIV, and it can only be men, and they need to be accessible for 8 visits. So that is a challenge to actually find. There are a lot of interested people that would like to participate, but when we tell them that during the next 6 months, you need to show up 8 times, that's the difficult part. But when you do the first-in-man clinical study for this indication, where we're actually aiming to wipe out all cells carrying HIV virus, it has never been done before. Everyone else is going for the virus and inhibiting the replication. We are trying to kill all the cells by the immune system. So I don't have a good estimate, because it's up to the patients. And as a CEO, I cannot even participate in that part according to good clinical practice. So our PIs are proposing for every patient that comes in to participate. But we realize it's taking too long, and we are, of course, eager, because more than half of the patients, we have the results, but I cannot see them before everyone is done. So we are as eager as you. So that's why we are also including more sites that can contribute, so we can get the final patients in.
Okay. So tight inclusion criteria,
Yes.
that's one obstacle or a challenge. And the fact that many people can live now with the medication that's available, is
Yes.
that also a factor in this?
Yeah, it is a big factor, because they feel healthy. So, by coming 8 times to the hospital, you start to feel sick again. I mean, no one wants to meet the doctor, even if they're nice, and get needle pricks. But in a study that is a first-in-man, you need to have a tight regimen, because we don't know, and safety is always our concern. So that is the drawback, that there is a lot of interest, but when you need to participate so frequently. So next study, of course, when we know much more, doesn't have to be so frequent. And if we get the results that we hope for, I think it's going to be the opposite, because when you can demonstrate that actually a few patients, we have eliminated the virus, we will have a lineup instead.
Yeah, and of course, clinical proof of concept will be a huge value inflection point for the company as well.
Yes.
And as far as the financing of these studies, I mean, you're in the HIV study, and then you are aiming to go into hepatitis B study, and as you said, starting then hopefully next year.
Yes.
Financing is always a challenge for companies of your size and so on. And this December, you negotiated a loan that will take you up until June of this year. Is that correct?
Correct.
Yes. And what will happen then?
Yeah, I mean, fortunately, we have a lot of options. We have very, very tight discussions now going on. I mean, not at least to say that some of the compounds we have may actually be useful for the coronavirus. So we have a lot of discussions going on, and I will be happy to share that later when I know more. But we have many options, including, of course, to ask the market again.
All right. We have a question here. Please use this microphone. Welcome.
How will you positively conclude that the HIV infection is eradicated, since these patients will still be on ART treatment?
It's actually very simple. You measure the amount of HIV integrated DNA, which is actually our primary endpoint. So we will measure in extracted CD4 T cells, which are the carriers of the HIV virus, and ask, is there any HIV DNA? And if it's none, there you have it.
Then it's a jackpot.
If you're lucky, but
If you're lucky, yes. But I mean,
otherwise-
even a decrease in HIV viral load will be a huge success because it has never happened before. So we hope that this is going to be the beginning of the end of the HIV virus.
how long will you follow up the patients?
The patients will be followed a total of 6 months, so 3 months on medication and 3 additional months follow-up. So we do not know if 3 months is enough of treatment, but that was what was available from the ethical committee in the discussion with the MPA. So I mean, tuberculosis, another chronic bacterial disease, you treat for 9 or 12 months. So it might be that we get a signal that shows the right direction. Perhaps you need a combination with checkpoints, or you need a longer time for the treatment.
Thank you very much, Ola, for
Thank
your
you.
time. And you're going to be hanging around here for the break as well, so people can grab you in the hallway.
Yes. Thank
Okay.
you. Thank you.
Thank you much.
Thank you.
Warm hand to Ola.